In this episode of EHA Unplugged, we will revisit a spotlight talk from EHA2024. Dr. Krzysztof Szade from the Jagellonian University in Krakow discusses hematopoietic stem cells in health, aging, and malignancy with a focus on metabolism.
During my last years of research, during PhD, postdocs, and when starting my own lab, we focus on hematopoietic stem cells, HSCs. Especially, we look at their heterogeneity and how they age. And they age in quite specific way. So, the first thing, with time, this reconstitution potential of HSCs is decreasing, but actually the numbers of HSCs expands. And what is very typical for them, they show so-called myeloid bias, so they are prone to differentiate more to myeloid cells. So there were different models that explain this. One assumes that this is just the epigenetic shift and driven gene expression. The other says this is the clonal selection model. So assume that there is like a subclones that are intrinsically have this myeloid bias differentiation program, and they just are selected during aging, and you end up with the myeloid bias. Of course, you can think about the composite model of these two, and we'll get back to it today. So I want to summarize our results, our findings in 3 different settings of HSC aging and highlight the metabolic context of it. So first, the stress-induced aging. So we know that under pressure, in stress conditions, we age faster. So this is the same with HSCs, yes. The second law is called physiological aging. So you may live in comfortable conditions, but you won't escape aging. And finally, I want to show how, at least in short, how the HSC looks when there is a malignancy in the system. So let's start with this premature aging. It happens that I studied an enzyme called heme oxygenase 1, HO1. So HO1 degrades pro-oxidative heme into the anti-inflammatory products and generally thought to be cytoprotective enzyme. So we look on HO1 knockout mice and actually see that already in the young animals, there is expansion of the HSC pool. So then when you look on peripheral blood, you already see a general myeloid bias there. And finally, when we transplanted the purified HSCs, we observed there was reconstitution. There are the main symptoms of HSC aging visible already in young HO1 mice. So we thought that this represents some kind of premature HSC aging model. And it seems that it is really, even at broad transcriptional level, the young HSCs from knockout animals cluster together with old wild-type HSCs. The HSC from young HO1 deficient mice showed already a premature aging. So what about the metabolism here? When we do gene enrichment analysis on this young HO1 knockouts among HSCs, one of the first hits was pyruvate metabolic processes. So there was a beautiful paper by Dr Szade lab showing actually there is so-called glycolytic metabolic stemness signature. And this depends on pyruvate crossing. So pyruvate can be put to the mitochondria by PDH for the OXPHOS respiration, or by transfer by LDH to lactate in glycolysis. And this glycolytic score, this glycolytic path here defines this metabolic stem cells that they show. And but it's also important, there are the PDKs. So PDKs can inhibit PDH by phosphorylation, and by this suppress the glycolytic metabolites influx into the mitochondria. And in fact, this is the PDK expression is regulated really much in transcriptional level, so it gives you opportunity to see so-called pyruvate crossroad gene signature. So we check how it looks in this HO1 deficient HSCs, and in fact, this is the case. So they downregulate LDH and PDK2, and it seems like this pyruvate in this HO1 knockout HSCs goes more to the OXPHOS. But actually, we were able to measure the ATP production among the strictly defined populations, and you observe that they don't use this OXPHOS because there's actually lower level of the ATP inside this H1 deficient HSCs. This glycolytic metabolic stemness that is dependent on this pyruvate crossroad is disturbed in this premature aging model of HSCs in this H1 knockout animals. So the next question was, is that the case also during the physiological aging? But at the time, people already knew that there is some heterogeneity among HSCs, but there was no prospective marker to dissect this heterogeneity and study it during aging. So first thing what we did, we screened for markers that are very specific only to HSCs, and among these markers, to find the genes that are differentially regulated during the aging. And this is how we found the neogenin 1. So majority of HSCs in all the mice expands, and majority of them are neo1 positive actually. The new one negative HSCs do not expand with aging. So then we see what is the difference between this new one plus and new one negative HSCs. The new one plus upregulates some cell cycle genes, but the most important for us was that they do express genes that are typical for mature myeloid and platelet cells. We have the hint of this clonal selection model myeloid bias that can prospectively study this. So, but of course the function is the most important here. When we transplant 10 cells, HSCs new one negative or positive, and indeed the new one positive HSCs mostly produce granulocytes, while this new one negative HSCs provide more balanced output. But there was one more question to say if this is really the clonal selection model here. So the question was, is there any hierarchy dependencies between neo1 negative and positive, or they live independently one of each other? The proper answer would be, this is a composite model, because we have both clonality here, but also a hierarchy and genetic shift, with neo1 negative HSC standing in the apex of this hierarchy, differentiated and giving rise to neo1 positive biased HSCs. So the question was how the glycolytic metabolic stemness look here within this changing between young and old. To do so, we make an atlas of aging HSC people, so very, very early stages of this HSC differentiation. So generally you see the clones, you see also the epigenetic model, and how the whole system looks regarding the pyruvate. So in fact, there is no much difference during aging or neo1 plus and negative subpopulations. So at the end, I just want to show you the last model, so the model of HSC in malignancy. So this is a very hot topic right now. Then the pre-leukemic HSCs and clonal hematopoiesis is mainly studied in context of MDS or AML in elderly people. So we did a bit different thing. And so I want to show you our recent observation about HSCs, but in pediatric ALLs. We observed a very strange aberrant B-cell differentiation program that generally is typical only for B-cell progenitors, but we observed that the part of HSC already expressed the signature of B-cell differentiation. And what was interesting for us, this part of HSCs also express high glycolysis score. So with this I want to end. I would like to thank all of my lab members.